Please come downstairs. Waziha, Ghania, please come here. You have to submit the feedback form along with this questionnaire. So please complete the feedback form as well as this MCQ question paper. Question paper. बैठ MCQ paper. आपको नहीं मिला है? नहीं, नीचे नीचे बचे हैं बच्चे. आप बीच में चले जाइए. The time starts now. 30 minutes का पेपर है. लिखा है. 1 घंटे का पेपर है. ऑफलाइन ना 60 minutes. ऑनलाइन कम क्यों दिया? चीटिंग कर देगी. Everyone is requested to switch off the mobile phones. All the questions are compulsory. आप लोग नीचे आ जाइए. ऊपर जगह नहीं है, आप लोग नीचे आ जाइए.
For your kind attention, only 15 minutes are left and you have to submit your question, the answer sheet at 3 o'clock. You are also requested to submit the feedback form. These are the requirements that has to be met, then only your certificate will be processed. At 3 o'clock, we'll be starting the final lecture cum tutorial in which Professor Hetao will be explaining you the methods of fabrication of DNA nanoparticles, and he'll be showing you some videos of his own lab where his students will be designing different DNA nanoparticles. So, first of all, submit your copies on time, the feedback form on time. Thank you. Keep quiet. Please submit your copies. You have only 10 minutes. And those who have not yet collected the feedback form, please collect from the organizing team, fill it up, and return back. Feedback form is very important. All of you have submitted your copies, or anybody is still doing? Aisha? All have submitted over there? Done? Everybody? ठीक है. Count all the answer sheets. We'll be starting in 5 minutes, the next lecture.
First, second. I think we start at the 3. So.
एक मिनट मतलब मैं शेयर कर दूं उसके बाद
Please pay your attention. Welcome back. You have now just completed your test. You've just appeared in exam, an MCQ based test. The mandatory requirement for Gyan. Now we are starting, I'm very sad also, the last session of this Gyan program of the 5th day, the last tutorial which Professor Hitao, he has initiated. Now he is doing the, from Agaz to Anjam, he'll be taking the last lecture. In this lecture, he'll be explaining you about the preparation and characterization of nanostructures, the DNA-based nanostructure, and how they are actually being done in his own lab. So I welcome Professor Hitao Liu. Please give a huge round of applause.
Thank you. All right, so in the next 20 minutes or so, I want to share some practical aspects of DNA nanotechnology, and in particular, I asked one of my graduate students recorded a video of the whole process that we used in our lab, making the DNA triangle nanostructures that we have used in many of our experiments. You may recall in the second lecture today, I showed a lot of experiments, and a lot of those are based on this triangle structure that we have been using since 2010. Just to give you a quick refresh of what this is one of those structures. Let me turn on the pointer. So this structure is created. So just to give you a refresh, this is the structure created first by Paul Rothman, the guy at Caltech, who in 2006 invented the origami technology. This is done by mixing one scaffold strand, the MP13 bacteriophage DNA. The length of this DNA is about 7,000 bases long. Again, we know this sequence, so we use the computer program to come up with about 300 of DNA staple strands. Okay. And when we order this is coming from a commercial DNA synthesizing company called IDT, Integrated DNA Technology. And we essentially got 300 small test tubes, 300 of those tubes. Now, the unique thing about this origami approach of making DNA structure is it's highly robust in the sense that we do not care too much about the stoichiometry. So if you think about your typical, most of the organic synthesis or inorganic synthesis process, you really want to make sure, oh, I add exactly the right amount. Okay. That's not the case here. So instead, what we do is we have all the 300 different kinds of short staple strands. We mix them together at roughly 1:1 ratio. Okay. Sometimes the concentration is not quantified very well when you purchase this DNA from commercial companies. They just do a quick UV-vis quantification, and sometimes they don't even do purification. Actually, we can use DNA strands that are not purified at all, just fresh out of the solid state DNA synthesis, no purification, no HPLC, no nothing, and very crude mixture. And we can use that, and the synthesis would work out just fine. So all the staple strands are, 300 of them. We mix them into one stock solution. And all the staple strands individually, they are roughly 1:1 ratio. Okay. And then we have the scaffold strand. This is a very long, 7,000 base pair long strand. And we usually make sure that the staple strand overall are in excess. So you can be as aggressive as 1 scaffold to 10 staple strand, but it can be as low as 1:2 ratio. As long as the staple strand is more or less in excess, you're fine. Sometimes we even did 1:1 ratio, and that turns out to be fine as well. So again, this is a highly robust process. It does not care about stoichiometry that much. Okay. And the reason being, you have so many ways to fix the problem. Because you have 300 staple strands, imagine if one of them is not enough, it's okay because you already folded the structures 299 times. The missing one it's not going to create too much a problem. Okay. So that's the reason this self-assembly process is not sensitive to the specific and exact stoichiometry, and that's a very unique feature of this, a very beneficial feature of this origami approach. Okay, so that's our stock solution. And then we make additional buffers, and you may notice the student specified pH 8, and this turns out to be important. Now, first of all, the stability of DNA nanostructure certainly depends on pH. Now, what's more important in this case is when we deposit DNA nanostructure onto silicon, which is one of the most popular substrates, the deposition, the interaction between silicon and DNA nanostructure heavily depends on pH as well. DNA carries a lot of negative charge because of the phosphate group. You may recall a question in the morning, what is the interaction between DNA and the silicon oxide surface? That is by ion bridge mediated by magnesium 2+. And to have that ion bridge interaction, you have to ionize silicon oxygen, the silanol group on the silicon wafer. And that requires a high pH. So in this case, we used a pH 8, and that just worked out well. And if we reduce the pH, oftentimes we see reduced adhesion of DNA structure onto silicon wafer. You also notice specific reference to magnesium 2+ in this figure. Again, that is very important because when you make double helical structure, you are packing a lot of negative charge into a small area, small volume. DNA has this negative charge backbone, and you want to neutralize that kind of negative charge. And the best way to do that is to add a divalent ion, magnesium being one of the most often used. Okay. As I said just before, magnesium 2+ also plays this role of ion bridge to mediate the adhesion of DNA structure to silicon wafer. Okay, so once we have everything, we mix all these stock solutions in with the appropriate concentration ratio. And usually, just for convenience, we distribute the solution. Usually, this will be in a 1 ml or 5 ml Eppendorf tube. We put them into smaller ones because later step we need to do centrifugation. We need to do a lot of things that requires mass balancing. So usually we split up into 4, 2, 4, 6 smaller tubes. And then we put it into a PCR machine. And what we're doing here is we're gonna heat up the temperature above the melting temperature of all the double helical sections. Again, here we have about 300 different strands, and each strand is about 20 to 40 bases long. Okay. We heat it up, first of all, to about 95 degrees Celsius to ensure complete denaturing of all the strands. And then we slowly cool it down. Now, the cooling rate is very important here because you want to give the system enough time to explore all those different reaction pathway. If somehow it got trapped in the wrong intermediate step, you want the system to have time to come back. So it's important to do the cooling slowly. Depending on the particular nanostructure you're making, this can go as short as a couple of hours to as long as 2 days. Again, the slow cooling would allow the system to get out of the trap state and come to the final thermodynamic minimum, which often is the nanostructure we're looking for. Okay. So once we're done that, what we end up with then ideally would be a lot of DNA nanostructures. And if you keep in mind, our staple strands are in excess. Okay, so we already have anywhere from 2 times access to 10 times access, depending on our particular mode of the day, and that actually fluctuates quite a lot. So at this step, once you cool everything to room temperature, you're going to have a lot of DNA structure, but also a lot of staple strands floating around. So you ideally would consume all the scaffold strands, but because you have access staple, so you need to get rid of those excess stable strands by some kind of purification. And in this case, we have some structures that are pretty big, 100 nanometer in size. You know, this is the edge length of the DNA triangle. Okay? And we also have a lot of single-strand DNA, essentially each about 20 to 40 bases long. Those are much smaller. Even if you're not considering the single-strand DNA are in random coil structure. Okay? Each base is about 0.3 nanometers if it's a double helical DNA. And if it's random coils, the effective size is going to be even smaller. So if it's double helical structure, 40 bases long is about 12 nanometers in length. But if it's a single strand, it's going to be random coil. It's going to be a random globular thing. That's probably a couple of nanometers in size. Okay. So what we do here, then we need to separate things simply by size. There are a lot of ways you can do the separation in molecular biology. You can easily separate biomolecules by size using gel electrophoresis, and that can certainly be done. But it just takes a longer time, and more instrumentation involved. You have to make a gel, and you have to stain the gel, and you have to extract the DNA extract from the gel. It's a lot of hassle. So the easier way to do this is to use a filter. And what you see here is this special filter. And at the bottom of, you see this is a 2-compartment filter. Okay. On the top part, we will add the DNA nanostructure raw reaction mixture to the top. And at the bottom here, there's a membrane that is size-controlled membrane. And you can pick a particular molecular weight cutoff. But because the size of the two components are so different, it's pretty safe as long as you pick something meaningful. Okay. So I think we really pick a molecular weight cutoff around 100,000 or something like that. And then in this configuration, the small species will go through. The DNA structure will stay on top. Okay. So to force the filtration happen, we centrifuge the whole thing at about 6,000 RPM for about 5 minutes. And we have to do this multiple times because we do not want to centrifuge everything to dryness. That will result in the DNA structure being stuck to the filter. So oftentimes we would centrifuge to about 1/3 of volume left. Then we dilute whatever is left with buffer and do it again. And then centrifuge again, dilute it, and dilute again. We usually do it 3 to 5 times depending on how much DNA, single-strand DNA do we want to get rid of. Okay. Most of the time, 3 times is sufficient. But in some special cases, when we really want to lower the concentration of single-strand impurity, we do it 5 times or more. Okay, so after that, we will collect whatever is left on top. Again, the filtrate contains the single-strand, excess single-strand. But the remaining liquid here contains DNA structure. As you see, there are triangles here and there. So we take those, and then the next step is we're going to do quantification. This is a standard technique everybody uses in chemical biology. DNA has a UV absorption, and then we simply do a quick UV-vis absorption measurement. And from the absorption data, we can quantify the concentration of DNA nanostructure in the solution. Once we have those, we usually break them apart into even smaller tubes so that we can store them in the fridge. If we store in 4 degrees Celsius, this usually survive at least a couple weeks, maybe even longer. We didn't push that limit very hard. If it's a room temperature, at least a couple of days, maybe a week. Again, we usually use them right away, but storage in fridge 4 degrees Celsius is not a problem. There are also a lot of research done to analyze and understand how this DNA structure would react to frozen conditions. And the general message there is, by freezing the buffer, you will do some damage to DNA structure, but not all. So there are also tricks to protect the DNA structure from the mechanical damage that occurred during the freezing process. And after that, we deposit the DNA structure onto a substrate. This usually silicon because it's flat. A lot of times we also use mica. Again, this is a very flat substrate. The height of the DNA structure is about 2 nanometers, so you really need something that is super flat at the atomic scale. And silicon wafer and mica are the two most often used substrates. In both cases, there's ionic interaction between the DNA structure with the substrate, and we'll be able to use atomic force microscope to image those. And so this is the whole process, and I'm going to show a video that essentially goes from step 1 all the way to step 10. I'm going to pause the video to explain what is what, so hopefully you'll get a more intuitive idea of, in the research lab, how this was done. Okay. And by the way, this was taken from a qualifying exam presentation of my student. His name is Shubhanka Kundu. And he also took the video that I'm going to show you in a sec. All right. Okay, so the person in the video, her name is Katie. She is an undergraduate student conducting research in my lab. She has been working with Shubhanka in the past 2 years or so, working in the area of DNA nanotechnology, broadly defined. She is standing in my lab. As you see, my lab is very disorganized, a lot of chemicals sitting around. But on the positive side, she has gloves. That's good. Chemical hygiene protection. She already has her glassware, so, and because she's working with ecosystem, I thought, okay, that's fine. And I'll play this and I'll pause from time to time. Most of the steps is pretty self-explanatory. Okay, but if you have any questions, feel free to interrupt me. Audio is gone.
And then we have pre-prepared DNA staple strands. And we combine those two in a 10-to-1 ratio, so 10 times the amount of staple strands. The other two things are water and some buffer, scaffold DNA or...
Okay, so I'll replay from the beginning. And as you see, she's explaining everything. So hopefully I don't have to interfere too much.
origami nanostructures. So it's a mixture of four things. First, we have DNA scaffold strands that we buy.
Now that scaffold strand, that is already a mixture of 300 different DNA scaffolds into one. So when we bought those scaffold strands, we have a pack, this array of Eppendorf tubes. And when I started, I don't have a lot of funding, so my student pipetted one after another 300 times to make a stock solution. But fortunately, we don't have to do it again because we already did it 15 years ago. We're still using that mixture until today.
And then we have pre-prepared DNA staple strands.
So this is a long, very long strand.
And we combine those two in a 10-to-1 ratio, so 10 times the amount of staple strands. The other two things are water and some buffer. And we usually combine those in a tube just like this using some micropipettes. And once the solution is mixed together, we divide them into 4 equal allocates into tinier tubes like this.
Okay, another reason we use the smaller tubes is because those are the standard. I think I'm not a chemical biologist by training, but I think those are the standard tube size for PCR machines. And because we use PCR machine for the annealing process, which is just convenient, we move them into the smaller test tubes, Eppendorf tubes.
And then we take them to our PCR machine. All right, so now that we have our 4 tubes of DNA solution, we bring them to our PCR machine, and we put them in the little slots right there. And then we get to select what type of nanostructure that we want to make. So there's a bunch of different kinds, but you just select it, press start. They can take anywhere from 2 hours, even up to 48 hours. And once that's done, we take all 4 tubes and put them into one centrifuge tube. Now these tubes actually have a filter in them.
Okay, so that's the size selective filter I was talking about. So as you see in the video, this part was placed on top of another Eppendorf tube. So the filtrate will naturally go down, and whatever is left would be would contain the DNA structure. And also another feature I want to mention is in some experiments, you will eventually be limited by the power of the PCR machine, because in some cases you have to do it really, really, really slowly. And in those cases, sometimes you have to be inventive and creative and come up with some kind of apparatus yourself. For example, a big styrofoam box so that you can cool it really, really slow. Because not all PCR machines will allow you to do 2 days or even a week of slow cooling. Sometimes it just doesn't exist. Okay. But in most of the cases, as she mentioned, it could be as short as a couple of hours. But we have done some other experiments where 2 days of slow cooling is required.
If you can see that. And as the solution gets filtered through via centrifuge, so we'll just go like this, only the DNA staple strands come through, while the DNA triangles, or whatever nanostructure we make, stays in the top section. And we add buffer as needed just to make sure that the solution doesn't run dry. And once we filtered it through, then we take it to our UV Vis nanospectrometer machine to check the concentration of DNA in solution.
Okay. All right. So another thing I want to mention about the purification process, because we do not want to filter, have the DNA nanostructure solution being filtered completely dry. So if you reduce the volume, if you centrifuge down to the volume of original 1/3, and then you refill buffer to the original volume, you're gonna reduce the single strand impurities strand, their concentration by a factor of 3, right? Because original volume is 1, you essentially remove 2/3 of them, and then you refill. So if you do this once, then you're going to reduce the impurity concentration by a factor of 3. If you do it twice, that would be a factor of 9. If you do it 3 times, that would be a factor of 27. Now, you may think this is quite a lot of purification, but you should also keep in mind that we're using huge excess of staple strands. As Katie mentioned, she's in this particular case, she's using 1 to 10 excess of DNA single strand, the staple strand. So ideally, after the reaction, you have 1 to 9 concentration, being 1 being the DNA nanostructure, 9 being the excess single-strand DNA. Okay. So if you do the purification twice, you will get concentration ratio of 1 to 1. So from that perspective, your reaction mixture is still very, very dirty. So in this context, you need to do the purification 4 times so that you can reduce the concentration of impure single strand to about 10% of that of DNA nanostructure. If you do it 5 times, then you can reduce it to about 3%, and that would be something I consider adequate, 3% impurity. Now, you can also, think about this at the very beginning of making this nanostructure. Instead of doing 1 to 10 ratio, maybe you can use 1 to 3 ratio, and that will change the calculation completely, and then you don't have to do the purification as many times. All right, so I think the next step she's going to do
our UV vis nanospectrometer machine to check the concentration of DNA in solution. Okay, so to check the concentration of DNA in the solution, we go to our spectrometer machine and open the software and hit nucleic acid. And once that loads, it has to do its own verification routine. And then at the top, we follow the instructions. So our blank solution will just be water. So I'll take 4 microliters of water.
So the instrument he used is a microvolume UV-vis absorption spectrometer, and what you see here, so this part, let me see if I can draw it. So this part, there's a sensor there. You can add a drop of water droplet onto it, and you push this part down, and that water droplet will form a meniscus, and it only requires a couple of microliter volume. So it's very economical in the sense that you are not wasting a lot of your sample. We also have other instruments that require the more conventional cuvette, but that requires much more volume of your sample. Now, when you think about the kind of volume we're dealing with, usually it's not even more than a milliliter. So, so we prefer this kind of micro scale measurement. Let me clear everything.
And I'll put it right on the tip where the sensor is. So now we have just a tiny little.
All right, you may see this is a small water droplet on top of the light sensor, and when the top of when there's this cap come down, it will form a defined gap, and from there you can quantify the concentration.
Droplet on there. We close the machine. And now that we've done that, we hit blank to measure the blank. All right, now it's saying we can load our sample. So first we're just going to use a Kim wipe to wipe up all the water so that it doesn't interfere with our concentration measurement. And so now I'm going to take a sample that we've already prepared and filtered. I'll take another 4 microliters of that and put it in the same place as the water. All right. We close the machine again. And now we click the big green button. And so our goal after we create a sample is to have a concentration between 20 to 50 micrograms per milliliter. And so as you can see, Right here in this green box, the concentration is 46.8.
Okay, so you may find this is a familiar shape. If you have taken chemical biology or biochemistry, this is a familiar shape of UV absorption of DNA, and you can quantify the concentration by looking at absorption at 260 or 280 or a combination of them. My memory is rusty, but the number here you can see A260 and A280. Those are two important numbers that chemical biologists usually use for quantification of DNA. From the absorption, we can calculate the concentration, and we want a certain range of concentration because most of our work are done by via surface supported, substrate supported sample. So we want to get a reasonable surface coverage. We don't want it too high or too low.
Which is just fine for us to store in a fridge to use later. Now, if the concentration was too high or too low, we would just take the solution back to the centrifuge and either centrifuge it more or just add some more buffer until we get the concentration that we want. Back in the lab, after we check the concentration of...
Okay, so now she's sitting in our shared facility and she's operating an atomic force microscope. And she's gonna show the sample loading and the data collection process. I believe the sample she's investigating is on a silicon wafer substrate, or could be mica, but one of the two. So I'll stop and explain what the machine is in a sec.
Our nanostructures, we would take them and put them onto some type of substrate, let that deposit, clean it off, and then bring it down to our AFM room. So here's some examples. This one right here is a silicon wafer, and the rest of these are mica. Those have our DNA nanostructures on them already. And then next, we have to set up an AFM tip. So here's a box of these tips, fun fact. This whole box is $2,000. So we have to be very careful with how we handle them. That's why right here we've already got the tip set up in...
So this certainly depends on the manufacturer of AFM. So different model of AFM have different procedures to load sample, to load tip. So this particular model is a Xylem, Xylem MFP3D. And it's a pretty good instrument for standard topography imaging, but still is a 10 years old model. We have been using, you see the Asylum Research, that's the manufacturer of this, this AFM. And this is their tip loading setup, I believe.
In here. And once that's ready, we can take our sample and the tip to the machine. So how does the AFM machine actually take an image of our sample? Well, let's imagine my hand is our sample, and this is a DNA nanostructure. And so the tip that we talked about before would be lowered onto the sample, and once we start taking an image, it vibrates like this and moves along the sample. And once it gets to some sort of structure, it moves around it because it doesn't actually touch the sample, but just hovers right above it. So if there's anything in the way, it has to go above it. And that's how the instrument actually takes an image.
I want to make a small correction. It's tapping, but it's actually in a non-continuous contact with the sample. There is actually a non-contact mode of AFM imaging, but that's not the imaging mode we're using. We're using the more common tapping mode imaging, vibrating the tip at about 300 kilohertz in our particular setup, and having the substrate moved up and down so that the amount of tip sample interaction is always kept at a constant. So this big box is the AFM sample chamber, and inside is the scanner and the sample holder and everything. There's electronics outside the box. As you see, there's a lot of wires and stuff. The whole box can also be purged with nitrogen. As you see, there's some kind of gas cylinder regulator here. In this case, we are not particularly concerned about the atmosphere, but it's possible to purge everything under nitrogen, for example, if that is needed.
And because the tip is so sensitive, it's very sensitive to vibrations and sound, so it's in a fancy little box.
So a little bit more on this instrument. The whole thing is put on a vibration isolation desk. But even on top of that, you see this big chunk of rock, just to make it more isolated from building vibration. And this whole lab is also on the first floor of a very old building. So the vibration is minimized in the first place. You also see the inside, a lot of aluminum foil and things like this. That's designed, I believe, to reduce thermal fluctuation because we are measuring a lot of dimensions at the nanoscale. So that's 10 to the minus 9. Now, if you think about the thermal expansion and contraction of a lot of materials, changing the temperature by 1 degree or even less can produce a lot of thermal contraction and expansion. So you have to create a very stable environment in terms of temperature, airflow, and vibration, certainly, to get high quality imaging.
So we would take our sample and this, put our sample onto the glass slide here.
Okay. So as I said, the way it works is the tip will scan the surface. Now that's the easy way to explain it, but in reality, what happens in this model is that the tip actually doesn't move. The sample moves. Okay. So the tip stays at the same location. There's piezo, so you see this is a sample holder, and there are piezo drivers in both X and Y dimension, and also actually Z. So when the tip vibrates, it just vibrates its stationary location, and the sample moves back and forth in a 2D scanning pattern.
Then we take our little thing with the tip and put it into this part of the machine.
And this part, that's the scanner. And it holds the tip, and she will very soon put the scanner, so right now it's upside down, and she will invert it so that the tip will be facing the sample, and then she'll do other things to make sure that tip eventually will contact the sample.
To make sure it's secured, then we take this very carefully, put it on top, and so now the wafer and the tip should be right on top of each other. And once this is set up, we go back to the software to set a few parameters. So after setting those parameters in the software here, we close the door to the machine, press go, and the image will show up in these boxes over here. And so this would be a 20 micrometer by 20 micrometer square picture. And once it shows us that, we can zoom in and out in different locations, which will end up looking something like this.
Okay, so she's not showing the real-time operation. So in a real operation, you would need to first load the tip, approach the tip to the surface. Now the tip is vibrating all the time, and as the tip is getting closer, the degree of vibration would feel being interfered, and the machine can detect that. And once it detects this smaller vibrational magnitude, the machine knows, oh, it's hitting the surface. And once that happens, it will maintain. It will stop pushing even further to the surface, and instead using the piezo in the Z direction, to maintain a constant tip sample distance. And then the scan will start. Some important parameters we typically use, one is the scan rate. This is how fast you scan in one back and forth. Okay? And in a typical experiment, we usually do a 1 Hertz scanning. This is actually pretty slow because you go here and then go back. So let me draw this. So what does this 1 Hertz mean? It means I go this direction and come back. That's 1 second. Okay. Now, each one of those images, you'll also see other parameters, scan lines and scan points. And those are 256. So what this means is each image here is made of 256 lines. And in each line, the machine takes 256 data points. So the resolution of this image is 256 by 256. Okay. Now you can increase that resolution or decrease it as your experiment would require. Usually we do 256, 512 is also common. You increase the resolution, but you will pay price in time, because if it's 1 Hertz scanning, then each image will take 256 seconds to be collected, and that's about 4 minutes. So a lot of the time of my students spend on sitting in front of the computer and evaluate the sample and find the correct sample area, so on and so forth.
So that was 20 micrometers. This is a 3 micrometer section. And here are our DNA origami triangles, which are just one of the structures that we're able to make. And so you can see this triangular shapes here, three straight sides, and you can see the whole.
So the important feature we look at here is the triangle, but also there's a hole in the middle. And this particular structure is very, very useful for a lot of our lithography work. You may wonder why we have been using this triangle structure for so many years. And the reason is because it's very, very good for that kind of work. So in this seemingly very simple structure, there are a number of features that are very useful. One is the sharp edge. Another is the hole in the middle. And even actually, there's additional smaller holes on the edge of the junction of the edges. So all those features would allow us to make a very quick and qualitative quantification and understanding of the quality of pattern transfer. That is the most important parameter we are trying to understand and improve. So for that reason, we've been sticking to this triangle structure for a long time, and we're most likely going to keep using them in the foreseeable future.
In the center of the triangles, and what this this lets us know is that the structures were made correctly. However, you can also see some of them aren't quite perfectly triangular. Maybe some of them don't have a hole in the center, or maybe the sides just came apart. And that's what we're able to check in the AFM image.
Okay, so that hopefully gives you a general sense. Sure. I'll play again without interruption so that you have a more meaningful understanding of what's going on.
Hey guys, today I'm going to show you how we prepare our DNA origami nanostructures. So it's a mixture of four things. First, we have DNA scaffold strands that we buy. And then we have pre-prepared DNA staple strands. And we combine those two in a 10-to-1 ratio, so 10 times the amount of staple strands. The other two things are water and some buffer. And we usually combine those in a tube just like this using some micropipettes. And once the solution is mixed together, we divide them into 4 equal allocates into tinier tubes like this. And then we take them to our PCR machine. Alright, so now that we have our 4 tubes of DNA solution, we bring them to our PCR machine, and we put them in the little slots right there. And then we get to select what type of nanostructure that we want to make. So there's a bunch of different kinds, but you just select it, press start. They can take anywhere from 2 hours, even up to 48 hours. And once that's done, we take all 4 tubes and put them into one centrifuge tube. Now these tubes actually have a filter in them, if you can see that. And as the solution gets filtered through via centrifuge, so we'll just go like this, only the DNA staple strands come through, while the DNA triangles, or whatever nanostructure we make, stays in the top section. And we add buffer as needed just to make sure that the solution doesn't run dry. And once we filtered it through, then we take it to our UV Vis nanospectrometer machine to check the concentration of DNA in solution. Okay, so to check the concentration of DNA in the solution, we go to our spectrometer machine and open the software and hit nucleic acid. And once that loads, it has to do its own verification routine. And then at the top, we follow the instructions. So our blank solution will just be water. So I'll take 4 microliters of water, and I'll put it right on the tip where the sensor is. So now we have just a tiny little droplet on there. We close the machine. And now that we've done that, we hit blank to measure the blank. Alright, now it's saying we can load our sample. So first we're just going to use a Kim wipe to wipe up all the water so that it doesn't interfere with our concentration measurement. And so now I'm going to take a sample that we've already prepared and filtered. I'll take another 4 microliters of that and put it in the same place as the water. Alright, we close the machine again, and now we click the big green button. And so our goal after we create a sample is to have a concentration between 20 to 50 micrograms per milliliter. And so as you can see, right here in this green box, the concentration is 46.8, which is just fine for us to store in a fridge to use later. Now, if the concentration was too high or too low, we would just take the solution back to the centrifuge and either centrifuge it more or just add some more buffer until we get the concentration that we want. Back in the lab, after we check the concentration of our nanostructures, we would take them and put them onto some type of substrate, let that deposit, clean it off, and then bring it down to our AFM room. So here's some examples. This one right here is a silicon wafer, and the rest of these are mica. Those have our DNA nanostructures on them already. And then next, we have to set up an AFM tip. So here's a box of these tips, fun fact. This whole box is $2,000. So we have to be very careful with how we handle them. That's why right here we've already got the tip set up in here. And once that's ready, we can take our sample and the tip to the machine. So how does the AFM machine actually take an image of our sample? Well, let's imagine my hand is our sample, and this is a DNA nanostructure. And so the tip that we talked about before would be lowered onto the sample, and once we start taking an image, it vibrates like this and moves along the sample. And once it gets to some sort of structure, it moves around it because it doesn't actually touch the sample, but just hovers right above it. So if there's anything in the way, it has to go above it. And that's how the instrument actually takes an image. And because the tip is so sensitive, it's very sensitive to vibrations and sound, so it's in a fancy little box. And so we would take our sample and this, put our sample onto the glass slide here. Then we take our little thing with the tip and put it into this part of the machine to make sure it's secured. Then we take this very carefully, put it on top. And so now the wafer and the tip should be right on top of each other. And once this is set up, we go back to the software to set a few parameters. So after setting those parameters in the software here, we close the door to the machine, press go, and the image will show up in these boxes over here. And so this would be a 20 micrometer by 20 micrometer square picture. And once it shows us that, we can zoom in and out in different locations, which will end up looking something like this. So that was 20 micrometers. This is a 3 micrometer section. And here are our DNA origami triangles, which are just one of the structures that we're able to make. And so you can see this triangular shapes here, three straight sides, and you can see the holes in the center of the triangles. And what this this lets us know is that the structures were made correctly. However, you can also see some of them aren't quite perfectly triangular. Maybe some of them don't have a hole in the center, or maybe the sides just came apart. And that's what we're able to check in the AFM image.
Okay, so I also brought some actual samples from Pittsburgh. Now, those samples,
Hey guys, today I'm going to show you how we prepare our. This is box is $2,000. And bring it down. So here's some examples. This one right.
Okay, so those are exactly the same samples she's using, and I'm just gonna show this around. Now, keep in mind, what is on this sample is tiny DNA structures. You can see them with atomic force microscope, but unfortunately, they're not visible to human eyes. Now, I wish I could bring the AFM here and show you everything, but before I become super rich, that's not going to happen. So there are two kinds of samples. One is a silicon wafer, that's a small piece. And then another sample is a mica glued onto a metal disc. The metal disc simply make it easier to handle the mica piece. So I'll just walk around, and if you're interested, you can take a look. I'm also going to walk in front of the camera so audience online can take a look. I'll give the mica sample to Dr. Kang, and he can show it around as well. I think that's pretty much what I want to talk about. Be happy to answer any questions. Um, any questions either about the current session or everything I have discussed in the past 5 days. It's been a
In the
great
past
pleasure.
5 days also, you can ask any question. Some students were asking which is the best substrate, whether it is silicon wafer or mica or maybe some other.
That's a great question. So both silicon and mica are quite often used. Mica usually, in my experience, usually give better quality of the image because mica is by its own, due to its own structure, atomically flat. Mica is a layered compound, and you can do exfoliation. It's also cheap. So mica has been a common and popular substrate for atomic force microscopy imaging for many years. We like silicon wafer for a lot of other reasons, in part because we've been developing lithography technique specifically for silicon. That's the reason we've been using silicon. But if you are just doing characterization of DNA structure, chances are you'll find better result on mica substrate. Any other questions?
Any other questions?
All right. If no more questions, again, thank you for your presence and attention to everything.